Hello, my name is Professor Matthew Schmidt, and I'd like to welcome you to genetics. In this session, we're going to talk about chromosomes and cell division, and we're going to have a particular emphasis on the cell division process called mitosis. Chromosomes, let's give a little bit of historical background and sort of catch up to where we were. Remember, Mendel was very good at doing crosses and collecting data and describing how traits were passed on from parents to offspring. However, he really had no idea what his genes or factors were made of, where they were located, and nor could he. It's okay, I'm not criticizing him. However, it turns out that chromosomes are the carriers of the genetic information, and we're now going to try and look at how chromosomes behave, what they are, and try and integrate that material with what we already know from Mendel's work. So here's a rather beautiful, I think you'll agree, picture of some chromosomes. These are chromosomes, all of them sort of look like an X, and we'll see why in a moment. They don't all really look quite as symmetrical as this. This is a bit of a simulation. But when we envision them and when we see them under the microscope, they do take on this type of an appearance that looks like an X. They really are quite beautiful. So, just to recap, Mendel's laws certainly seem to be true. They seem to be consistent, and all was well with the world in terms of the Mendelian world. But as I just said, sometimes we want to pretend for the sake of argument that we don't know things. But at this point, almost everybody, even non-scientists, know that genes are in fact made up of DNA. He had no idea about that. He also did not know they were located on the chromosomes in the cell. The major thing that his work did allow was a prediction of the outcomes of crosses, assuming that you knew, dominance relationships and things of that nature. So Mendel's work was rediscovered around 1900, and just about that same time, a discovery usually attributed to a scientist named Sutton was that the genes of Mendel were in fact located on chromosomes. Now, chromosomes are structures that are visible under even a reasonably powerful microscope, and they're located in the nuclei of eukaryotic cells. So peas and humans are both eukaryotes, at least we have that in common. Now, guys, it was known that chromosomes existed prior to this just because cytologists who looked under microscopes saw them. But the connection was never made that these chromosomes and the genes really were very, very intimately connected. The way he did it, Sutton, and there were other people involved, basically is he looked at the way chromosomes were moving during cell division. And this is something we're going to talk about today. And what he realized is that they behaved exactly in the same way that Mendel's laws would predict if Mendel's factors, what we call genes, were in fact located on these chromosomes. So it's almost looked like the chromosomes were the factors in a sense. We'll see why, but basically this was the first time you could say that genetic analysis and cytology is just the study of cells, looking at cells, came together so we could make this really important connection about not just how the information behaved, but where it was and what it was to a point. And I also just wanted to mention that, now we know, in essence, it's a little oversimplified, but chromosomes are really just long molecules of DNA that are condensed to various degrees. So it's going to take us a while to get to the molecular part, the DNA part, but for now, let's think about chromosomes. And we're not going to continue to emphasize it here, but keep in the back of your mind that this is goes along with what Mendel was describing, even though it's not strictly about the information itself. The chromosomes, now we're looking more at their structures and how they behave. Okay, so we are going to focus on cell division today, and one of the reasons is because think about this. We talked about in the pea plant experiments and even in our human pedigrees, information was being transmitted from a parent organism to its offspring. Now, that's reproduction, but cell division is a form of reproduction, and I dare say all forms of reproduction involve cell division one way or the other. Think about it this way, and maybe it'll be clearer. If you were a single-celled organism, well, you probably wouldn't be watching this video right now, but if you were a single-celled organism like a protozoan, the cell is the organism, right? And if that cell divides, we'll talk about the mechanism, you now got two new organisms. That is reproduction, right? And traits are being passed from that parent. It's not exactly the same, obviously, as the way we do it, from the original cell into the so-called offspring cells. So Mendel could maybe predict the way the traits would go and the ratios. Now we want to see how cell division actually works, which is sort of a reinforcement of how the information is flowing, given that the information is located on these structures. So some terminology that we need to be aware of. Adult eukaryotic organisms, whether that be you or I, whether that be an adult pea plant, we have a diploid chromosome number. You may be familiar with the term diploid. Algebraically, diploid is 2n. We'll discuss that briefly in a moment. But that's the chromosome number. So in humans, 2n happens to equal 46 in humans. That really is not a magic number. Humans happen to have 46 chromosomes. Pea plants, it turns out, have 50. There's really no real rhyme or reason to the exact number that any particular organism has. But it also means you can look at our complement of 46 chromosomes in two different ways, I think. One of those ways is to say that we have two complete sets, and those sets, each one is called a haploid set. It doesn't really come from this, but I think of sometimes haploid meaning half of the total. It's not just any half, but it's one complete set. In other words, if we use humans as an example, through your father's sperm, you received one complete set, and through your mother's egg, you received another complete set. So one complete set is algebraically known simply as n, and that gives you the idea that our diploid number, 46, is the combination of these two haploid complete sets. So I hope perhaps it's obvious that if 2n equals 46 in humans, n would equal 23. Okay? So that's one way you can think of it, is that our 46 chromosomes are really two sets of 23, two complete sets. However, I mean, it doesn't change what they are, but there's another way you can think of it, and that is that we have 23 individual pairs of chromosomes. Okay, because what happens is within the set, we number the chromosomes arbitrarily based on their size, 1, 2, 3, 4, 5, 1 being the biggest, 22 being the smallest. So another way of looking at this is that you have two chromosome number ones, perhaps obviously since they're part of the set, but you got one number one from your father, one number one from your mother. And the two number ones are similar in a variety of ways. So they're referred to as homologous chromosomes or homologs or even homologous pairs because they are a pair. So I guess my bottom line is you can think of it as having two sets of 23 or 23 pairs of homologs. It is what it is, as they say, but those are two different ways to envision it. So those two members, we're going to see that the homologs, sometimes it matters that they're homologs, sometimes it's not particularly that important. Now, in sexually reproducing organisms, which are almost all animals and plants, in fact, many animals and plants can reproduce either asexually or sexually, but Mendel's work focused on sexual reproduction. So let's use again humans as an example. There are two fundamental types of cell division.